Good morning, everyone. My name is Wendell Lim. I'm a professor at the University of California, San Francisco. And it's a great honor to be able to moderate the first scientific session of this really tremendously exciting meeting. In many ways, it's very fitting that we're going to start with hearing from the people that in many ways will shape the future of the emerging field of synthetic biology, namely some of the participants in the annual iGEM competition. So for those of you who aren't familiar with iGEM, iGEM stands for the International Genetically Engineered Machine Competition. So every year for the last couple of years, groups of students from universities around the world have worked over the summer to design and execute projects in synthetic biology of their own design. In November, they gather at MIT for the iGEM Jamboree. So that's what's depicted here. As you can see, last year there were over 50 teams from all around the world participating in the Jamboree. For those of you who haven't been to the Jamboree, I have to tell you it's really the most exciting meeting that I've been to. You can really feel the excitement and energy of these young people who truly understand and believe in the power of learning how to design and reprogram biological systems and what that can do for us. Last year, among the 50 teams that were participating, there were 5 teams that were chosen as finalists for the grand prize, and we're lucky to have representatives from 2 of those teams here today. The first team that we're going to hear from is from Peking University, or Beida, which won the grand prize, and we'll hear from Shili Zhu and Chunbu Lo. And then the second team that we'll hear from is the team that I was an advisor for, the one from UCSF. One thing that's unique about the UCSF team is that it's one of the first teams that was primarily composed of high school students, particularly high school students from one of the local public high schools in San Francisco. And we're going to hear from Alex Eng, who was a member of that team. So let me turn it over to Shili and Chunbo, who from Peking University, who are going to speak about designing a push-on, push-off genetic circuit.
Thank you. Thanks the organizer for giving us this honor to share our work with you. Chunbo and I present Peking University iGEM Team 2007, and the title of our talk is Modular Design of a Genetic Push-on, Push-off Switch. Modularity is defined as a system can be separated into functional subunits. Those subunits can keep their function and recombine to a new system. Just like the wall E constructed by Lego bricks, it can be separated into eyes, wheels, clouds, and so on. Each element has its own function, and if we put them together in a different fashion, we can get a new robot as YF or G. Biology is just like Lego games. It's modularized in DNA level, protein level, as well as network level. The functional modules can be shuffled during evolution process, and generate the system has new function or behavior. A classic way to build a synthetic biosystem is to bottom-up assemble the functional modules. The method is emphasized by iGEM. System in each level can be considered as a module and used to build a larger system. To construct a synthetic biosystem, we need to face 2 problems. First, how to build a functional module, and second, how to couple those modules. We built a genetic push-on, push-off switch as an example for the idea of modularity. By push-on, push-off switch, we mean the system can give sequentially alternative output and the same input. Just like the cartoon shows, we push the button, the lamp is on. And push the button again, the lamp is off. So on and so forth. We put the genetic push-on, push-off switch into E. coli and use UV radiation as input. Green and red fluorescence are used as output. Assume the bacteria is green at first. After UV radiation, it turns red. UV radiation again, it turns green. So on and so forth. Intrinsically, the push-on, push-off switch is a sequential logic circuit. It can give output based on both the input and the internal state. The circuit must have two main modules. The memory module remembers the previous state, and the combinatorial gate, which integrates both the input and the memory. Also, the combinatorial gate can give a feedback to the memory module. We use the NOR gate as the combinatorial gate. And the input passes the inverter to get to the gate. Both the input and the feedback of NOR gate are transient pulses. The input has an additional function that it can clear the memory module. It makes sure the memory module can accept the feedback of NOR gate only when input comes. Technically, the memory module has two parts, a clearable memory and the memory of it. The two parts have different time scales. The signal passes the memory module as a wave. It makes sure that the signal from input and the feedback of NOR gate will not mix up. The NOR gate is 1 only when the memory of memory and the inverter are 0. Otherwise, it's 0. Here is the state transition table. Assume initially the state of memory is 0. When there is no input, the inverter is 1. Based on this table, the NOR gate is 0. When input comes, the inverter is 0. The clearable memory is cleared, and the memory of it is still 0. Based on this table, the NOR gate is 1. When input gone, the inverter is 1. The clearable memory gets feedback from the NOR gate, and the memory of it changes also. Based on the table, the NOR gate is 0. When input comes again, the inverter is 0. The clearable memory is cleared, and the memory of it is 1. The NOR gate is 0. When input gone, the inverter is 1. The memory module learns from the feedback of NOR gate, and the NOR gate is 0. So on and so forth. The input is transient pulses, and the output reports the state of the second part of memory. So we can see the circuit behave as a push-on, push-off switch. We also build an ODE model. The system can behave as a push-on, push-off switch under a set of suitable parameters. Based on the logic circuit, we designed the genetic push-on, push-off switch. The promoters, transcription factors, and other basic elements are well modularized, so we can take them directly from Nitro system and rewire them under our purpose. First, we designed the memory module. A bistable switch serves as the memory module. It's composed of a positive feedback and double negative feedbacks. There are two promoters. PRM controls the gene expression to the left, and the PR controls the gene expression to the right. MRFP and the EGFP are used to report which promoter is on. And two transcription factors. C1 activates PRM and inhibits PR. When C1 exists, there is only gene expression to the left. The bacteria is red. C1434 inhibits PRM. When C1434 exists, there is only gene expression to the right, and the bacteria is green. Technically, PR is stronger than PRM, so the green state is dominant. Then we design the NOR gate. A double reprise promoter serves as the NOR gate. It inhibits by two repressors. One is LEX-A. It exists universally in the cell. And the other one is LEX-I. It's controlled by the memory module. P0 is on only when neither of the repressors exists. C1 induced minus is controlled by P0. It activates PRM and inhibits PR, just as C1 does. When C1 induced minus exists, it will help the cell to set up the right state. When UV comes, it activates the protease REC-A. REC-A exists universally in the cell, and the activated REC-A can cleave C1, C1434, and LAC-A specifically, but has no influence on LAC-high or C1 induced minus. To describe the dynamic process, we divide the system as 3 parts: gene expression under PRM as part 1, under PR as part 2, and under P0 as part 3. Initially, since PR is strong and PRM is weak, part 2 gets transcribed. C1434 inhibits PRM and further stabilizes the green state. Meanwhile, P0 is repressed by LAC-A, so there is no C1 induced minus transcript. When UV comes, C1434 and LAC-A are cleaved. P0 is on, C1 induced minus transcript. It activates PRM and inhibits PR. So part 1 gets transcript and part 2 is silenced. After uridation, P0 is repressed by both LAC-high and recovered LAC-A. And the C1 substitutes C1 induced minus and maintain the right state. When UV comes again, C1 and LAC-A are cleaved, but P0 is still repressed by LAC-high. So there is no C1 induced minus transcript. After uridation, C1434 is transcribed by the strong promoter PR and set up the green state. And finally, LAC-high will dilute it during cell growth, so the cell returns to the initial state. That's how the switch behaves as a push-on, push-out switch. So that's all the introduction part, and my work is done. Chunbo will introduce our experimental data. He will show how we construct the system and how the system behaves.
Okay. Thanks, Xili. I will introduce our experimental work. To realize our design, we employ 2 strategies: rational design and direct evolution. In biology, the biostable switch have been extensively studied, so we have enough information to design it as a memory module. It's similar to a NOR gate, but it's not easy to design the interconnect part because the on and off between different modules usually don't match well. First, we design the memory module. It's known that a positive feedback or double negative feedback of dimerized transcript factor can generate a biostable switch. Besides the dimerization, the transcript factor can bind in DNA with high-level cooperation. After using our mechanism, we help our biostable switch to be a stable memory module. After transforming the plasmid of biostable switch, we got this colony, as shown in the picture. The picture is taken by stereoscope and merge the green and the red fluorescence channel. Each round spot shows a colony which grows up from a single cell. Each cells growth are connected. It means no jumping happens during cell growth. So we got a memory, right? This slide shows the green state is dominant as we. Then we design the NOR-gate module. In order to obtain a big region between on and off state of NOR-gate, we insert a consensus operator into a strong promoter. Furthermore, we also elaborate aside the inter-operator distance. Previous results show that when the distance is 19.3 base pairs, the repression level reaches maximal. We follow this principle in our design. Our measurement shows the gate function well. The repression level is high only when both repression is low. We also got the NOR gate. Okay. After I got the two function module, we couple them by direct evolution by fine-tune the two interconnected parts like I and the C1-induced minus. We vary the two paths repression level by mutating their ribosome binding site, RBS, because RBS is modular and easy to change. We choose the strongest RBS as a backbone and mutate their key point. In here, we develop a screening method for the direct evolution. After construct the two mutation library, we transforming them into E. coli and select the colony taking both plasmid, and we check the colony's color and the stereoscope and pick up the green colony into 96 well planes and incubate them for selection. Then we titrate the exponential growth culture to on two planes, to same planes. One plane is exposed to UV radiation, other is not. Overnight growing, we pick up a red colony on the UV triggered planes. Then we incubate the red cell in the 96 well planes again, titrate the culture, and triggered by UV. Finally, we pick up the green colony on the UV triggered plane. After the two rounds of selection, we got a 3-strain reward. Following the 3 successful strains, our pictures are taken by stereoscope. Each spot shows a cell colony. At first, the major population is green. After triggered by UV, the major population turns to red. We pick up a red colony and triggered by UV again. The major population turns to green. The other strains have similar behavior. In order to make sure this switching is caused by our design circuit rather than stochastic jumping, we do several control experiments. This control focuses on decouple our submodules. First, we delete like I. The string can switch from green to red, but not from red to green. Because without like I, C1-induced minus always express under UV. It helps the cell set up a red state, right? Then we delete the C1-induced minus. Without the help from C1-induced minus, the string can switch from green to red, but it still switch from red to green. Finally, we delete both like I and the C1-induced minus. This string also can switch from green to red, but switch from red to green. It's similar to the second control because both of them lose the output of the NOR gate. After UV clear the memory part, as Shili introduced, no matter what the previous state is, the green state will dominant as we designed. With this control in mind, we conclude that the designed circuit give output based on both internal state and the current input. It's a typically sequential logic. As we know, the UV is a toxic to cell and can cause the cell to death. So in the future, we can replace Y-type REC-A with a constitutive REC-A mutant. For example, the REC-A 730 food activity not depend on UV radiation. Thus, we can control the REC-A mutant and the inducible promoter and change the harmful input to other survivor ones. Thus, we can get a new genetic module. What we can do with this module? Here we give a sample. Such module can be used to construct a genetic counter. If we have several proteins with different, like REC-A, with different recognizing site, in here we show the module 3 count. Of course, we didn't want to build a supercomputer with this some modules, but it's still possible to implement some interesting function. For example, the function of a Kruger machine. Here show a Kruger machine. If the machine learned that you have insert your money, you can get the cook when you push the button. Otherwise, no matter how hard you push the button, nothing will happen. This work is partially implemented by PKU iGEM. After the jam break, we continue it in our labs. We would like to thank the PKU iGEM 2007. We spent an exciting time together last year. We also would like to thank our supervisor, Professor Oh Yang, who is really supportive to our work. Thanks Professor Tang, Professor Lai, and other members in the Center for Theoretical Biology. We also thank the 10 lab member of 10 lab and the colleagues in UCSF who have a lot on the presentation. Thank you for attention.
Okay, thank you very much. That was terrific. Yeah. So, do we have any questions? Yes, back there. Can you you said, did you sequence?
Yeah, did you sequence your evolved strain and observe any changes to the sequence which you designed?
Yeah, sure. We sequenced that exactly as we designed, and we also sequenced the SD mutant.
And were there changes to your design sequence that were better that you hadn't anticipated?
Oh, you mean if we change our design, it will be better or?
No, after the directed evolution step.
Oh, after the direct evolution, we sequence the successful strains we got. We know the mutant of the RBS. The 3 strains are different, but we didn't measure the strength of the RBS.
But the mutations were limited to the RBS.
Yes.
Okay.
Thanks.
Other questions? You can use this.
I really enjoyed listening to your talk because everything was so familiar. PL, PR, C1,
Yeah.
etc. And my question is, because that's since 1970, I mean. So my question is now, are you going to remake all the elements instead of sticking with poor old Lambda, whatever nature design, or are you going a scratch, make new promoter, everything the best?
Because it's design, so we must, based on previous result, the lambda switch is started very well, so we adapt them. If we have this element, many system we can adapt other system.
That's okay.
Sorry.
It's a good question, whether we should stick with the parts that nature has given us or make better ones. But I think we'll hear a lot about that in a lot of the talks during the meeting. Other questions? So how many times can you push the button?
We push it once and pick up the colony who change the color and push again.
So that means twice?
Yeah,
Okay.
Only twice.
You see the picture after we trigger it from green to red. Actually, the major population changed green to red, but there is still some cells stay in green. So the switching from green to red is not ideal. So I think if we trigger it more times, maybe that will mix up. So we'll improve that later.
Yeah, that's why I asked the question. I wonder, as you push it, your population.
Yeah, I understand.
At some point, you'll lose it, right?
And another thing is, UV irradiation is a harmful signal, so it will cause some toxin to the cell. So if we give UV irradiation so many times, finally the cell will die. That's why we want to change the wild type REC to a mutant that not depend on the UV irradiation.
Great, thanks.
How long is the conversion time?
Based on the UV response, because UV response is more than 2 hours.
Okay. Other questions? No. Okay. Well, thank you very much again.